high-risk populations, such as immune-compromised individuals, will benefit from such a multi-pronged approach. In fact, in July, analysis of data from the phase 1 study of CM04S1, published in the peer-reviewed journal iScience, showed that CM04S1 demonstrated potent and equivalent T-cell cross-reactivity against Delta and Omicron variants. These findings suggest that T-cell immunity stimulated by CM04S1 may constitute a critical second line of defense to provide long-term protection against SARS-CoV-2 variants. Now, I'll repeat, these were based upon the analysis of data from the human clinical trial phase 1 study of CM04S1, which was indeed published in iScience. CM04S1 is currently being evaluated in 2 phase 2 clinical trials. One trial is a comparative study of CM04S1 as the primary vaccine versus the current FDA-approved Pfizer vaccine in people that have received or are undergoing specific blood cancer therapies associated with transplantation or CAR T therapy that suppress or severely reduce pre-existing immunity to COVID-19 vaccine. Multiple clinical evidence has demonstrated that such patients fail to respond optimally to the current generation vaccines, and we believe that the CM04S1 will prove to be more potent because it is multi-antigenic, delivered using the MVA vector. We believe this will differentiate CM04S1 from the other vaccines by providing both a strong antibody response and a sustained T-cell response in these patients who are still at high risk of severe COVID-19 due to their immunocompromised status. The other phase 2 trial underway is evaluating CM04S1 as a booster for healthy patients who have previously received either the Pfizer or Moderna mRNA vaccine. We believe that providing a heterologous booster rather than a continued multiple shots of the same vaccine or similar vaccine may provide more robust and durable immune response protection. Heterologous prime boost immunizations are well studied in other fields such as HIV and are being evaluated in multiple countries using different COVID vaccines. Finally, the ongoing Govx effort to develop an advanced MVA manufacturing process based on a continuously growing avian cell line to increase production consistency and capacity will well mesh with the clinical development activities and full development schedule associated with the CM04S1 and the CM02 vaccine. Not alone in recognizing the limitations of the current authorized COVID-19 vaccines. The continued need for more durable immunity, the waning of protective response, insufficient protection among various high-risk immunocompromised populations, and other evident issues are well documented and of increasing concern. We applaud the federal government for realizing that significant incremental funding is needed in support of next generation COVID vaccines, providing the promise such as our CM04S1 and our CM02, as well as other vaccines, therapeutics, and technology. This was underscored by the actions of the Senate Appropriations Committee's recent FY23 Health and Human Services Appropriations Bill, specifically targeting next-generation COVID-19 vaccine development. We look forward to continued dialogue and discussions in support of CM04S1 and CM02 as we continue to advance the development of these important COVID-19 or coronavirus vaccines. The WHO declared monkeypox a public health emergency of international concern. Nations worldwide are enacting procedures and policies in support of minimizing the health risk from monkeypox to their populations. Currently, there are 2 vaccines authorized in the U.S. for prevention of monkeypox, the primary vaccine being modified vaccinia ankara, or MV, which is also the vaccine utilized in numerous Govx vaccines, including our CM04S1 and CM02, which target COVID-19. In addition, MVA is the vaccine vector used in our hemorrhagic fever virus vaccines against Zaire Ebola virus, Sudan Ebola virus, and Marburg, as well as our development stage Zika virus vaccine and even our MOC1 cancer immunotherapy. In fact, previous peer-reviewed publications addressed the successful prevention of monkeypox in non-human primate models following the administration of Govx MVA-based HIV vaccines. Recognizing the global public health need and attention of monkeypox, evaluation is underway related to CM04S1 and the prevention of monkeypox. It is anticipated that the results will demonstrate successful protection, validating that CM04S1 is protective against both COVID-19 and monkeypox. We also anticipate validating our hemorrhagic fever virus vaccines as protective against monkeypox, potentially providing unique vaccines preventing both hemorrhagic fever viruses and monkeypox in a single vaccine. This would be very important in certain endemic areas of the world. We look forward to reporting more on this topic soon. Now I'd like to turn the presentation over to Mark Reynolds, Govx Chief Financial Officer, for a review of our recent results and financial status. Thank you, Mark.
Thank you, David. Starting with our income statement, I'm going to go through some of this pretty quickly. I know most everybody wants to get to the Q&A. But starting with the income statement, I'll focus on the comparative figures. Actually, that's our balance sheet first. Okay, here we go. Income statement. Grant revenues were $82,000 in the 6-month period of 2022 versus $190 in 21. That's reflecting a wind down in both our grant from the NIH supporting the COVID-19 vaccine and the grant from the U.S. Army supporting our Lassa fever vaccine program. As of June 30, 2022, all the currently available funds for those grants have been utilized. We do intend to seek additional non-dilutive funding for our development programs in the future. R&D expenses were $2.6 million in 2022 versus $1.4 million in 21. With the increase as expected, primarily associated with new clinical trial activity for COVID-19 and the cancer programs, and that includes manufacturing costs for clinical trial materials. The increase is also reflective of higher personnel and consulting costs as we staffed up for an overall higher level of activity. G&A expenses were $2.1 million in 22 versus $1.8 million in 21. With the increase also associated with higher personnel, consulting, and higher patent costs. So overall net loss for the first 6 months of 22 of $4.7 million, or 47 cents per share, versus $2.9 million in 21, 49 cents per share. Again, with increase primarily associated with ramp up of organizational infrastructure and other costs associated with CM04S1 and conducted clinical trials. Going back to the balance sheet, our cash balances at June 30 were approximately $31 million as compared to $11.4 million at the end of 21. And as David mentioned earlier, the change in the cash balances were reflective of $8.2 million used in operating activities, offset by proceeds from our stock offerings in January and May. The combined net proceeds was nearly $28 million. And these numbers, I'll point out, also do not reflect the additional $5 million came in just this week from warrant exercises. So current cash stands at about $35 million. Funding our 3 ongoing phase 2 clinical programs and preparing for the next stages of development are the most significant use of our cash and our top financial priority. And as I noted, we just received $5 million through the exercise of the warrants. And based on that, our outstanding shares now stand at 24.7 million. That's a current number. So in summary, we are well positioned to accelerate and advance our clinical programs with a cash runway sufficient to fund our operations and priority programs toward the end of next year. And I'll be happy to answer any other questions during the Q&A, but I'll turn now, call back over to Dick.
Thank you, Mark. My colleagues and I will now answer your questions. Again, joining us for the Q&A session are Drs. Mark Newman, Kelly McKee, and John Sharkey, our Chief Scientific Officer, Chief Medical Officer, and Vice President of Business Development, respectively. I'm therefore turning the call Back to Andrew for instructions on the question and answer period.
Begin the question and answer session. To ask a question, you may press star then 1 on your telephone keypad. If you're using a speakerphone, please pick up your handset before pressing the keys. To withdraw your question, please press star then 2. At this time, we will pause momentarily to assemble our roster. First question comes from Jason McCarthy with Maxim Group. Please go ahead.
Hey guys, thanks for taking the question. Can you guys talk a little bit about the dynamic around the need for a monkeypox vaccine? Because we know NBA works. You would be, I think, the most advanced US-based NBA player base, and then we're seeing some of the mRNA companies try to nose in Moderna today, coming out and saying their stock is up sharply. Can you give us a little bit of color around those dynamics? What that space could shape up to be, or is it really just NBA and you guys could be significant players there?
I'll start, and then we'll see if any of my colleagues would like to. But first of all, Jason, thanks for your question. Appreciate your interest in the company. We do recognize that we more than likely are the leading MVA company, certainly in the United States. We all know that there is a single source supplier for monkeypox currently. That's out of Denmark. I think it's very important for there to be a supplier out of the United States that addresses this for numerous reasons, not only to reduce the total dependence on a single supplier. I have no idea to the degree to which Moderna with their mRNA may or may not be successful in producing a monkeypox vaccine. Time will tell on that, but we certainly recognize that MVA, which as we know is the basis for so many of our products, is approved for preventing monkeypox. So that provides us an opportunity, and we validated that in previous peer-reviewed publications that I mentioned in my comments. Now, I don't know if Kelly McKee would like to add anything else to that, but I'll ask if he would. Kelly?
Yeah, hi. David, I think you sort of summarized the sort of overriding considerations. MVA is, or at least vaccinia-based, or vaccinias in general, are really the only proven preventive measure for monkeypox at this time. And the regulatory pathways for new entrants, I think, is yet to be decided. So how much Moderna or others will be able to play in this, I'm not sure anybody really knows at this time. As we look to how we would position ourselves for monkeypox, we've got to deal with some regulatory questions as well. So it's kind of a wide open field right now.
So do you take a multi-pronged approach, meaning you have what looks like a really good COVID vaccine that could also potentially cover monkeypox, but you also have Sudan and Marburg and Ebola, and monkeypox might be more of an issue in other countries. Would you consider doing something here in the States and maybe something in like West Africa? We saw another group announce they're going to do something in Kenya just this week.
The answer is yes, and I'll ask Kelly to elaborate a little bit about our outreach to Africa and the concept there, and also the difference in monkeypox and the strain of monkeypox that we see in Africa. Kelly, would you like to address that?
Sure. So Jason, I mean, your thinking aligns very well with ours as well. The potential to offer sort of a twofer is certainly there for us, and we have made some preliminary inquiries with to individuals in some of the sub-Saharan African countries to explore interest in some sort of a co-development program. I'm not at liberty to sort of disclose any in the nature of those discussions, but suffice it to say the potential is certainly there. You know, the strains of monkeypox, there are two strains, predominant strains of monkeypox, serotypes of monkeypox, that have been recognized. The one that's causing sort of the global epidemic right now is a West African strain, while the Central African strain of monkeypox, which is predominant in Central African Republic and sort of neighboring countries, is a much more virulent. It seems to be much more virulent than the West African strain, and so I think there's a lot more concern in these potentially endemic countries for having access to an effective vaccine, and the opportunity to offer protection against both monkeypox and another endemic virus, be it COVID or one of the hemorrhagic fever viruses, is certainly an appealing prospect.
And just last question, what about interest from a group like BARDA, where the stockpiling could be? Because, I mean, the monkeypox virus, the MVA ones they supply, as you know, I mean, it's for smallpox or immune-compromised people. Like, does that make your COVID vaccine for immune-compromised people that has cross-protection against monkeypox particularly attractive to a government agency like that?
I would say we think it does potentially. We know that various federal government agencies are recognizing that the multi-antigenic approach for COVID-19 is something to seriously be evaluating and reviewing. So we know that from discussions, et cetera. And we also believe, based similar to what you've just raised, and that the having that, and which is of increased interest as we see these continued limitations of the current authorized vaccines related to COVID-19, but also then having the added benefit of being able to address monkeypox could be very important to the stockpile program, as well as to NGOs who are looking at other endemic areas.
Good. Thank you for the questions, or taking the questions out.
Thank you. Okay.
Again, if you have a question, please press star then 1. The next question comes from Jeff Frost with Crystal Research. Please go ahead.
Asked several of my questions, but on that same wavelength, if you will, the combination, there were stories around this week talking about the military in Africa, as well as talking about our military. You've already had Army grants. Is that something where, one, you would be able to actually supply that, something you would pursue? And secondly, have you talked with, and this goes down the pharma line, obviously, even companies like Pfizer and Moderna with the COVID vaccine were not able to supply up. Have you been able to move far enough down that line to be comfortable that with these going through, or even the monkeypox one going, that you will be able to supply? And the last question is, strength-wise, obviously, if you have a combination vaccine, that's great, as long as the government or the regulatory bodies want to put through a combination vaccine. Are you thinking about doing it, offering it separate as well as combined?
To answer the second one, as has been commented on, there's a complex. I would say this: we are reviewing. It's a very complex development regulatory pathway regarding to MVA as an alternative to the existing MVA that's out there. So for us to go forward and bring that forward, what we recognize is that our MVA-based vaccines have that added benefit of preventing monkeypox. So if we have an MVA VLP or MVA-based Zika virus vaccine, and we were to further that in development, let's say in a collaboration relative to the Southern Hemisphere, South America, we know that, for instance, in Brazil, that monkeypox is fairly common and is also a threat. So there we would be able to meet the needs, not so much from I'll call it dual indication, but from the recognition that the MVA-based vector brings the benefit of also preventing monkeypox. So that's sort of the approach that we're looking from that. From the standpoint of manufacturing, I'll ask if Dr. Mark Newman would comment on that, because as I mentioned in my comments, we continue to make progress on moving towards a system that will enable us to meet the demands at a much faster rate than in the traditional chicken eggs or chicken embryo fibroblasts. Mark?
Yeah, sure. So right now we manufacture using a process based on chicken embryonic fibroblasts, which is the same thing that Bavarian Nordic uses. So it's a primary cell line, and that has limitations, and you start with eggs. We have an active program looking at getting into continuous cell manufacturing, cell-based manufacturing. And this is the more comparable to what the adenovectors. So if you think of the J&J or the AZ COVID vaccines, those are produced with these continuous cell lines. And so that's ongoing. Now our focus has been on our vectored vaccines, particularly CMO4S1, and then some of the other products that are at the research level. We actually haven't looked at just producing MVA without one of these inserts in it. So go to your other point, would you make something that was MVA specific? That would be the easiest path, I have no doubt. There's a lot of data out there that suggests there are multiple cell lines that can produce or support the production of MVA. Where we run into a little bit of our difficult situation is when we have these combo vaccines, because you're incorporating an insert, you got to make sure that insert stays in there while you're producing it. So that's just to make an MVA vaccine for the cell line would probably be the easiest path. And we have
that's why I asked, because that's why I asked, because from a regulatory path, historically, the FDA has had some issues when try to do things combined, and I think your data is very clear and very convincing separate, and I just think it might be easier for you to get these, even though doing it combined and all the benefits of working against this one and working against that one and the whole platform, great. I'm just thinking about what would be easiest for out there and simplest for someone to understand.
Exactly.
Yeah, no, I think you're right. If it's an HIV vaccine, you'd have to get it approved for HIV, and then you'd expand your claims, showing that it also works against monkeypox. Yeah, I think that's what we would face. So yeah, those are all things that are being discussed.
Okay. Thank you very much for the answers.
Thank you, Jeff. Thanks.
The next question comes from Kumara Guru Raja from Brookline Capital Markets. Please go ahead.
Thanks for taking my questions, and shifting gears to oncology. What are you seeing in terms of enrollment, and also in terms of the animal studies that are being conducted in North Carolina? When can we get an update, and what kind of information can we expect from that studies?
Kumar, thank you for your interest, et cetera. Could you repeat your first question, and then we'll address the North Carolina?
Yeah, the first question is just the details about how the enrollment is going, what you are seeing there, what can we expect in terms of updates.
Okay. So I'll ask Kelly to discuss the Gedeptin and all, and then I'll ask Mark Newman to pick up on the development programs, including the UNC Charlotte. Kelly?
Yeah, hi. We probably shouldn't really talk about the enrollment dynamics at this time. We transitioned this trial to take control. I mean, we in-licensed the Gedeptin product a number of months back, and we've been in the process of transitioning the IND and expanding the trial from a single site to a multi-site study. And that has resulted in sort of a pause in the